Pragya? See, now you can use the pointer. That will show up. This is the microphone.
I will hold this. Hello.
We start. Feel free to have some snacks. Feel free to start with the question. Don't forget to evaluate that.
So, hello everyone. So today's my topic for presentation is metagenomic profiling. So first, I'll explain about what is metagenomic profiling is. So first, if you can see in this figure, it says like you are taking sample from different area, different environment like soil, water, or human gut or human skin, any hard surface, like any environment. Then it will go to the lab, like for DNA sequencing. The second process is DNA sequencing, and then meta alignment start to work. Like we need to figure it out, like what's the genome in that particular environment present. Like if the process it is a process of analyzing microbial community by sequencing their DNA directly from environmental sample. So by using this metagenomic profiling, we'll get to know like in particular environment which organism present, and what's the percentage of that organism present in that particular sample. So here is the process, metagenomic profiling. So first, the collection of different sample from different environment like soil, water, and guts, and then DNA extraction, and then sequencing. After sequencing, the data analysis, to know the DNA that is present in that particular environment, and what is their percentage in that particular environment is called metagenomic profiling. So why is it important, like why we need it? So there are too many microorganisms like virus, fungi available in this environment that are not able to culture in traditional way in labs. So that's why we are using DNA sequencing and try to figure it out, like what organism present in that particular environment. So application is health and disease, like you'll get to know like what microorganism causing disease, and environmental studies exploring microbial community in ecosystem. So you'll get to know like which microbial community present in particular area. So advantage, like it can work on known or unknown species, and it's not biased, like it's not like biased about if the particular organism cannot be cultured, it cannot be used via using metagenomic profiling. So that's the biggest advantage of the metagenomic profiling. And disadvantage is computational resource. It's the storage needs, because the reference genome is too much, like it's and if you are collecting a species from particular environment, the genome, it's also very large, so it's a computational resource, and the storage is their disadvantage. And uh, so uh, if we'll get the environmental genome, let me explain in different slide. First, let me explain the background. Like again, I said, if you can see in this is the organism can be cultivated in a lab, and you can say this one is uncultured microorganism. So we need to know this. We can know this by using metagenomic profiling. So metagenomic profiling bridge this gap by enabling the discovery of new microbial species, and it will give information about functional genes and ecological interaction between different organisms. So there are three methods that is used in metagenomic profiling. First is marker gene. The example for marker gene is motus metaphyllin that I'm going to explain in my next slides. Kamer. So Kamer is like will break down the DNA that we collected from environmental sample and try to get uh, their particular presence in reference genome. So that is Kraken. And alignment based is like we align read to reference genome. Reference genome, if you know what is reference genome, is the library reference genome that is given by NCBI. Like it's a let me explain in this way. So here is reference genome, right? We have too many reference genome, like about human and different viruses, different fungi, and so that are present in reference genome. And this is read that we have from environment. Like from environment, we have different DNA of different microorganisms, that is called read. So first in this process, in first tool metalign, we'll get a Kmer from each sets, and then it will decrease the reference after getting Kmer. We'll try to figure it out, like a few shared Kmer discarded will be taken in the overlapping, and more shared Kmers would be taken in this overlapping. And then by using this two factor, will make new subsets, new subset database that is used for alignment for this reads. So after getting this alignment, we'll try to figure it out, like what's the percentage of E. coli in that particular sample, or different organism in that particular environmental sample. So why we use this? Because meta alignment is like 5 minutes. It will give result in 5 minutes, as compared to naive algorithm alignments, like different alignment, like previous used tools. And okay, so it's gonna be different. It's gonna be viral sequence, bacterial sequence, and different microorganism sequence, not human or anything, but different microorganism reference sequence. We're gonna use it for our read genome. No, it depends. Like if you have any idea about the your environment, like what can be in that particular environment, so you can filter it out your reference according to your environmental sample. Yes. Yeah.
Kind of matching your mouth or something.
Yeah, that's what, like we refer some of that are discarded, like that's not similar in between both of them, and some we are taking that are completely same in both the genomes.
So it's perfect match.
Yeah.
Means
Yeah. So here is the meta alignment processing, like database pre-filtering that we do in this process, pre-filtering. And then we aligned it by using align reads to the filter subset of genome using optimized algorithm. And we balance by using that, we'll cut it down, we'll cut this reference, like we'll make the subset of database, right? So it will reduce the speed, like example, like if it's a 787 GB, it will reduce to 7 GB. Like it's very convenient, like it's very it will reduce size while doing that. And the limitation is, okay, so you are saying in this, like,
that's 787.
yes.
So like that's really good. And what is the percentage that you're losing information?
Okay, so I think it it will lose some of the information, but it's so it will take the similarity, like a similar gene, particular sequences from particular.
So if the loss, be at which stage? Intermediate stage, final stage,
It's gonna be in intermediate stage.
how about final stage?
So final is like we are taking the read alignment and then subset. So whatever we lose here, we lost it.
Will
But
it affect final results?
some extent, like if the species abundance is less, like if it's less than 2% or 1%, you might lose some of the features over here. So you won't get here like them. So it's very sensitive to low abundance species in that particular environment. So the second tool is Kraken. So Kraken is basically a very simple tool. Like it will use k-mer generation. Like if you have environmental sample genomic data, you will break down into k-mers like 20 or 30 length, and you will try to get you have reference genome as well. So you have k-mers from there as well. So you will try to match them, and you will, while matching, you need to see like LCA mapping, like the lowest common ancestor. That is like the closest known ancestor of all organisms where k-mer is found. So it's not like you are taking the particular species, but you are going to take the ancestor, like the oldest one in that particular species. That's how you will get the Kraken use. And it's again, the limitation is reference database. Like if you don't have reference database, the good reference database, you won't be able to get the result. So the third is Motors database data set. So it's completely like it's take like in our meta alignment, we are taking reference genome, and here we are taking a reference genome from uh, different database. Like if you'll see reference Motors, that's a reference genome. Meta modulus, that is the genome that we found from environmental sample, like after reconstruction. And MAG is also that, like we'll construct environmental sample genome into MAG, and SAG is single amplified genome, like it's isolated from particular area, and it's contain only one DNA genome. So we are taking all the database, and
So what is
MAG is metagenomic alignment genomes. That is, we are taking if we have data set from any environment, and we found any new species, any new genome from that particular data set, we'll add it into, we'll make a new construction that is called MAG, that is found from different environments. Assembly, sorry, yeah, yeah,
means we get the reads, sample.
yeah.
We don't do alignment, we don't have reference.
We
don't know
just,
their location. Just
okay.
trying to tag them next to each other if the last character similar to the first character in the next. So we attach them together.
Okay,
We try
yeah.
to do this for all reads until we get complete.
Yeah.
Then we call it a MAC.
Okay, even we didn't have the
The
reference genome. Okay, okay,
de novo.
okay. But in this single amplified genome, we have
It's isolated,
isolated
means all reads coming from
same
genome.
genome.
So it's okay to align them together or assemble them together.
Okay.
So the MAC have too many species.
Mhm.
Each read belongs to different.
Okay.
So we try to attach them
Then,
together.
okay. So this is data set, and we can from environment we can get any of let me explain here. So this is the steps to do Motors tool use. So first we make a database, like here we did. So that's the same process. Then read mapping, like you'll sequence read are aligned to the marker gene database. Whatever a read mapping is, like whatever you have in the environmental sample, you'll try to map that by using this three references. And then how you'll count the profiling, like what's the percentage of that particular gene in that environment, that can be count by marker gene coverage to estimate species abundance, and including uncultured microbes also. So it's very good for uncultured microbes, or the novel organism that's new for environment. So so meta file and 4, it's another tool that I used. So here is the steps. So it's also depend on marker gene identification. So it has 5.1 million unique marker genes that is derived from known and novel. Like novel is new species that discovered or that might not be discovered yet, but metagenomic assembled genome that is taken from environment. So by using read mapping, the same thing, we'll try to uh, whatever we have in environmental sample, we try to map with this data set and get the abundance via using normalized marker coverage or detection SGBs, where greater than 20% our marker are mapped. Like from this, so our genomic data that are mapped with this particular data set, so we'll mark it down and we'll get the abundance uh, by using read mapping. So again, its limitation is database dependence. That's completely depend on all the tools depend on reference data set. And if you are missing any reference genome from so you won't be able to classify which genome is present in uh, environmental sample.
What is it?
So okay, marker gene is like
The gene is coming from the genome.
yes,
Marker
is
something unique.
yes.
Identifier or species.
Okay. E.
That's why we use it for the there is one portion of the genome that species. Find a lot of reads mapped to that region, tell you
Then
something. Okay,
particular.
probably these reads coming from that genome. That's why we have high similarity with that region. Right?
That is good. So here are pro and cons of meta alignment, Kraken 2, Mortis 3, and Metafile 4. So as you know, it's very high speed and pre-filtering, and it can use on large data set with high accuracy, but it's also depend on reference genome, and it's limited to by alignment based methods and pre-filtering bias. So Kraken 2, it's I said it won't classify. Uh, this cannot classify lower species level. Like if any species in the environmental area that is present 2%, it won't be able to identify that particular organism. So it's a lower specific level sensitive, limited to reference genome data set, and it will struggle with closely related species. Motors, it's slow than other tools because it has large data set that depends on MAGs and SAGs quality as well. And Metaphylan, it's high accurate profiling, integrate MAGs, and high sensitive for it. It this one and this one cannot find low abundance species. So their drawback is they won't be able to find all the organism that is present in that particular environment. So here is the result from the paper. So F1 precision and recall, and they give a false positive number as well. So in this we will see like how many false positive are present. So according to that, Kraken perform very bad, but it's very fast for the large data set. So you can consider this as well, because if you want to, it's a critical of if you want to find like environmental genomes very fast, you can use Kraken, but it will give false positive value more than other three. So
What does
so false positive means like it will give if the species is not present in that particular sample, but it will show that is present in that sample. So that's not good. Like, so, yeah.
the F1 score? See, you are comparing speech level,
Yes.
level.
So this one mainly work on genus. Like species is like a basic unit, basic taxonomical unit. So these three works good on that level, but genus level it's like it's uppermost like the second most taxonomical unit. So metagenomic used on that. Like it will classify species as well, but
It
it
works
won't.
in all levels. You-
Or genus
agree
level.
on.
Okay.
Is this copied from
Paper.
or your result?
Yeah, it's paper. This all. This
Which
thing.
one?
Oh, I used meta. I think I used
Meta
Modus
Plus.
3. Yeah.
That's not fair.
Okay. Okay. I use same thing in my. Okay, I can use all of them on genus level as well. Oh, yes.
Either
Okay.
up to you. You can choose. All of them are flexible. What level?
Okay. That makes sense. I need to modify my results. So
Did they
it.
provide any reasoning why?
No. But I used from the paper, so yeah, I might be wrong. So so all the tools doesn't depend like they all are specific for particular use case. Like metaphylan will work good on human microbe studies, so because highest accuracy for human associated species. Motors will work on environmental sample like soil, water. So it will, because it will detect meta motors, that is new novel organism that are present in database. And Kraken, it's again speed critical diagnosis. It's very fast. So it can process 100 million reads in less than 5 minutes. So if it give a false positive also, that's fine. Like if it's reading is very fast for large data set, and if it's is if you need a speed answer, you can use Kraken. And meta alignment is mixed pathogen reach sample. Like so it will balance alignment accuracy with pre-filter for reducing false positive. Here are my results. So I worked on a small mock community, 5 bacterial species that has so 2 high abundance and 3 low abundance species. So how my model works? Again, Kraken will give more false positive value as, but F1 precision recall also decreased in my case, and meta alignment perform same as it is presented in a paper. And here is the conclusion. Like all tools all tools doesn't work on all the data set. That can be that can depend on a particular sample type, research question, and need for speed versus accuracy. If you need speed, you can use Kraken too, and if you need accuracy, you will use different other tools. Depends on your research question, and overall integration of computational method enable more comprehensive and accurate exploration of microbial diversity that support in health, environment, and biotechnology. And so I already said no single tool dominate all scenario. So Metafile 2 and Motus lead in accuracy for human and non-human environment. Kraken is unmatched for speed, and but against speed the accuracy will fall because it will show more false positive value. And meta align is breach alignment between KMR, and it will give robust accuracy with manageable runtime. Future work, we can improve database coverage, computational efficiency, like we can replace some of the method with some machine learning model that will perform well, and ability to detect novel species across all environment, like novel new species from environmental sample. These are my reference paper. Thank you. Any question?
I see those F1 scores under default, right?
Yep.
Supervises, isn't it too high? Like, all of them are so high. Not this one, research.
Yes.
Almost perfect. But I need that.
Yeah, you're right, but so as you are saying, it's too much, like 95% accuracy is yes. So as we said, reference genome, we need to decide like which particular species of you need. Like if you need viruses, you will try to get the reference genome, and against your sample environment genome, so you will get more accuracy because the data set is not that big. So you'll try to get KMR that particularly they will get aligned KMR, so you'll get more accuracy according to.
So how you?
Yeah.
How did you calculate? Use marking tool or calculate?
No, in this they already gave it, but I used benchmarking. So I used between them, you are saying,
No,
or
this number. You calculate it yourself or you use the benchmarking? There are lots of benchmarking tools can generate this.
so as my data set is low, I did it by just calculating like
So you have a ground truth and you know
yes,
what should be
how
there,
it
what
works.
should be
Yeah.
there.
And or are they just trying to make it faster or something like that?
So
After reaching like done?
not done, like they are trying to because nowadays species like microorganism gonna increase, so we need to make it computationally flexible, like it should not consume. Can you repeat the question, please? Sorry.
The question is, let's say we reach 95%.
Yes.
What are the pictures from? What is it? Every order is like, oh, they are doing this, but they are doing like slow. So I need to make it fast. Or is it more? What is the limitation in this field now? What are they trying to do?
So false positive thing, if you can get more of it, like if you'll. Yeah, that's a question. Like I said, if you want it, you can replace the KMR and different algorithm with machine learning models. So that will make it faster. Yeah, you can do that because it's taking 5 minute time. So if you'll use a machine learning model and all, it will decrease your time, run time. Yeah, it's not too much. Yeah, that's true.
So can we say that this is over?
No,
What do you think?
I don't think so, but you can answer it. I don't know. I don't know. I don't know.
Now, there are first database. We have a lot of good genome code, 4 terabytes, 5 terabytes. You have to process this in your computer. None of the tools, but they require at least loading 100 gig of RAM in one shot. And that requires like server class. Second thing is the compute power. Some of them do really alignment. Imagine you're aligning to got the entire passkey file, you align for every single genome of the data, 4 terabytes. Some of them no, just extract k-mers, try to match them. Some others try to match to marker genes. Don't include the genome at all. Don't care about the 4 terabytes. Just try to get unique regions from the genome and compare to only this. So there is diversity in the computation. That directly affects the accuracy. When I go for the full genome, you get the best accuracy. When I go for smaller region in the genome, it will be faster, but it will hurt. So now the trend either improving the computational power or improving that. I was still very far from achieving high accuracy. All the high results you are seeing here is way too things that you already know that you are done to. But when it comes to real life, it's like the sample from the environment, just from this table. You'll be amazed by the amount of species that are unknown. We don't have reference genome for them. So you'll see 50% of your reads unclassified. You don't know what groups are. Other 50% things that we know.
Fire to also do like have a lot of missing data that we need to fill out.
Yeah, that's one thing. So there's still a lot of work. Coming to LLMs and Gen AI, still like starting. That will help by not comparing to the entire data. Good train, speed of speed before, okay. For high confidence, belongs to that speed. Specify that. But these accuracy, these numbers are questioned. So we have a paper in Nature Method to evaluate most of these methods of real data. You'll see, like you'll be shocked. This 99% accuracy will drop directly.
It looks good on paper. The results that you have.
Yeah, if there are great reviewers, they will directly pick on this number. Especially when you compare at different level. Of course, you'll achieve high accuracy, but here very low accuracy. So different level of classification.
Yeah, like you select those.
Yeah, for example, if you say this is COVID-19, yeah, of course you'll achieve very high accuracy because COVID-19 easy to capture. There are some unique regions. But if you tell me which variant exactly of COVID-19, that's the lowest level. This is the you could really differentiate this variant and that variant. If you go to species level, yeah, this is SARS-CoV-2, of course, we know. Then what's next? At least 2,000. Right, that's a great. Don't forget to validate.